Hi everybody, my name is Nick, and today I'm going to talk to you about variant Creutzfeldt-Jakob disease. So just to start off, we're going to talk about prion diseases in general. They were first described by Hans Gerhard Creutzfeldt and Alphonse Jacob in the 1920s. Prions are abnormally folded proteins that have been found to be causative agents of various diseases in humans: CJD, variant CJD, iatrogenic and familial CJD, as well as Kuru. In animals, scrapie, bovine spongiform encephalopathy, otherwise known as mad cow disease, chronic wasting disease of mule deer, and transmissible mink encephalopathy. On the right side of the page, you're going to see a couple of different histology slides of why it's called spongiform encephalopathy. As you can see, this vacuole right here, this opening in the brain tissue, over time starts to develop what looks like a sponge appearance, and that's just the dying tissue in brains. Just a little background on proteins and prions. Proteins are chains of amino acids, usually around 300 amino acids in length. They carry out many vital functions within the body. DNA is transcribed into RNA, which is translated to amino acids, and these are then folded further into various proteins. The function of protein is determined by the composition of these amino acids, and its function is changed by the way it is folded. So if you have a normally folded protein and it becomes misfolded or unfolded or anything like that, it can completely alter its mechanisms and usefulness. Prions are pathogenic. They are disease-causing proteins that have been folded abnormally, and they can cause damage to the cells they are in, but further into tissues, organs, and eventually entire organisms. And they do so by causing apoptosis, otherwise known as programmed cell death, and they can continue this cascade of misfolding and misfolding to other and other proteins. And you will see here, this is a normal protein, and this protein can go off and misfold another protein, or a prion will come and misfold this protein into what looks like this, and you can see it's generally the same structure, but these green and red sections are distinctly different, from these alpha helices on the left side to these beta pleated sheets. Just a little bit more information about prions and proteins. In a normal healthy cell, proteins generally live out their usefulness. This can happen by just no longer necessary, just high concentrations of proteins that are no longer needed in a cell, but essentially a protein can be determined to be no longer useful, and the process of proteolysis can regulate these unnecessary proteins. Proteolysis involves various proteasomes and protease enzyme, and these prions have been found to be resistant to protease enzymes, these enzymes that break down proteins. So one of the mysteries behind these prion diseases is how is it that a normal protein can be transformed into a prion? This mechanism is still not understood as to how a protein can go from this to this, but it is still being researched. Here's just a couple of different types of spongiform encephalopathy. Kuru is found only in humans and transmitted through cannibalistic practices. On the island of Papua New Guinea, it was a common practice to eat brain matter, spinal cord, brain tissue of a deceased relative. And this is the only way in which it's spread. Prevention and education and the cessation of these practices have now resulted in no new cases developing. Bovine spongiform encephalopathy, otherwise known as mad cow, is thought to be the cause of variant CJD, but CJD has two broad branches. One is classic and one is variant, and classic is further branched into sporadic and iatrogenic. Sporadic being the 85% of classical cases affects 50 to 75-year-olds, whereas iatrogenic is around less than 5%. And iatrogenic can be caused by transmission of prion particles through surgical procedures, implants, and medical devices. Here's just a couple more Distinctions between sporadic and variant: as you can see, variant is much younger in age, whereas the population for sporadic is much older. They generally both have a pretty similar presentation. Mental deterioration is pretty common with both dementia-like or Alzheimer's-like symptoms. Variant is much more prolonged than sporadic, and the brain tissue impacted, 100% of variant CJD, 100% of them have found cerebellar accumulation of prions, where in sporadic it's only been 40%. And a thing to understand about this is these are two distinct diseases. They are both caused by prions, and there's very strong evidence to suggest that variant is caused by the ingestion of contaminated meat. But CJD is a little bit less understood. There have been genetic links to the familial CJD and the classical branch, as well as surgical equipment contamination, corneal transplants, and dura mater grafts. And although these are two very distinctive diseases, they are both completely fatal. Transmission will occur when somebody becomes infected with the inoculation of nervous tissue. There are no modes of transportation through droplet aerosolization, and a person must consume the meat of an infected cow, or in the case of Kuru, the infected brain matter of a deceased person. There has been a lot of scare and hype in the media every so often about these diseases, about the prevalency, and it seems that as long as you stick to the regular meat and you're not eating the brain of cows, even if the cow had bovine Spongiform Encephalopathy, you should be fine. It's a very low risk of contamination because that's not where the prions accumulate. They accumulate in the brains of cows. The diagnosis and treatment of CJD. So there is no actual blood analysis that's shown to be effective because there is no immune response associated with it. The prions are just proteins. They are not bacteria or viral particles, so they do not elicit immune responses. A definitive diagnosis can be made after a person is deceased through a brain matter biopsy. In 2014, there was a study that showed that prions could be found in the urine analysis, but the treatment of these conditions is just purely supportive and pain management. There's just no effective treatments available out there for people with CJD. Some drugs can be shown to decrease the amount of time, or is shown to increase the amount of time that somebody survives, but nothing will completely arrest the progression of these diseases. And then, so here's just a couple of manifestations of what does it look like when somebody has a CJD disease. They will have a rapidly progressing dementia with myoclonus, which are just the muscle spasms. Psychiatric and behavioral manifestations and changes, both paresthesia and dysthesia is just a tingling sensation. Dysthesia is associated with pain, paresthesia is without. Cerebellar ataxia is the inability to balance or move your extremities, so a lot of people with these diseases can no longer walk and are pretty heavily impaired. Changes to the EEG, the electroencephalogram, amyloid plaque on the biopsy have also been shown. And here you can again see this vacuolization that occurs. This one is on a sporadic histology slide, and this one is on a variant CJD slide. You can see it's just riddled throughout the brain tissue of those infected. Alright, well, thank you for watching. I appreciate it.